Hey guys. How are things going? Carmen, everything fine? Could you find it online?
Yep, all good.
Yeah, guys, I posted everything. I still didn't make presentations for unit 7, 8, 9, and 10, but I will later today. How is studying going?
It's good.
You're not complaining, I'm glad.
I've been writing all over my walls.
That's a good strategy. I used to do that.
I have paper everywhere.
That's what I did. I remember that. So just I never did that for microbiology. I did that for zoology, for systematics, because it had multiple species, genera, families, a bunch of things that I couldn't memorize ever. So I made like that's exactly what you just did. Yeah.
I don't know if it'll help, but oh well.
But here is the thing. I know that Dr. Stein will never ask people to memorize meaningless things. So as long as you understand, you will be good. Like there is no point really asking people to memorize each single detail. Even though there are details in those lectures, I mean, again, you have to memorize some of them, but as long as you understand the point, you will be fine.
Mm-hmm.
I mean, that's from the, I actually got a final version of exam now, today, like just a few minutes ago. So I still didn't go through it, but I will, and I think it will be okay. I mean, hopefully. I don't want to be too optimistic. So let's see. Okay, not many people are here, but maybe somebody will come. I mean, how many people we had yesterday?
12 or 11 students and you.
Hey, could you guys get a recorded review session that I converted to the YouTube video?
Yeah.
Oh God, I'm genius. Super. I wasn't sure. I wasn't sure. Am I going to be able to do that? Because for me, this is all new technology. And with my new experiment that I'm just running now in the lab, I wasn't sure how much my brain will be able to operate, but I survived and it was good. Anyway, so are you guys okay if we start and then whoever comes, and again, everything will be online. So how I share, yeah, I share the screen. Yep, I'm sharing the screen. You all, I believe you all can see this, right? Hopefully. So if you remember this study session, and that was, so in this study session, we covered dilution rate. And first part of this review session did have dilution rate, but I'm not going to go through dilution rate now. And the reason is simple, there are no dilution. Can you all hear me? I mean, I hear something in the back, but that's fine. So again, it's posted online. All those dilution rates examples are posted online, but I don't think that there will be, I mean, I don't know final version of the exam, I didn't look it yet, but I don't think you will be asked to calculate dilution rate. That's more for micro 315 than for 311. So I will just go through questions through units 4 and 5, which are protein translocation, protein mobilization, and stress. And again, I won't stay on details. You read these things, definitely it will be helpful, but don't freak out if you cannot memorize some meaningless name of the gene or something like that. So, unit 4. How are proteins mobilized? So, mobilization or translocation across the membrane. So there are two types, two basic types, translocation and secretion. So that's why I'm constantly correcting myself. So proteins are mobilized either through translocation or through secretion. Those are two main types. When we talk about translocation, we think of two main systems for protein translocation, and that's SEC and TET. protein secretion is something different, and we will come to that point in a few. So the first system for protein translocation is SEC. And SEC translocation system has co-translational and post-translational version. So let's go first through co-translational. So we all know what is translocation, so no need, what is translation, no need to go through that. So in translation, protein is synthesized on ribosomes. Ribosomes are machinery for protein synthesis. So while protein is synthesized, so during translation, this protein is getting N-terminal signal sequence, and that signal sequence is a key. So if you get a question from this, I believe that it will be focused on meaning and significance of N-terminal signal sequence. So that's important to know. So again, protein now has N-terminal signal sequence. So this sequence is actually labeling protein for specific translocation. Now, one ribosomal protein is called SRP, or signal recognition particle. This SRP is recognizing this N-terminal signal sequence, but protein is not completed yet. Again, I wrote it. Yeah, somebody had a question? Sorry, I think I heard somebody wanted to ask something. You just stop me if I'm too fast, okay? Anyway, so again, I wrote it here. Some species have trigger factor, molecule that will have interaction between SRP, ribosomes, and proteins. So trigger factor is like a connective thing. So not all species have it. Anyway, so we had protein with N-terminal signal sequence, and we have SRP, which is recognizing that N-terminal signal sequence. Now, all complex protein in the middle of translation and ribosome, so all structure will be recruited to the membrane. So that's why it's co-translational. So everything migrates to the membrane. What's happening now on membrane? So in the membrane, you have a protein sitting and waiting for all this structure to come. That protein is important. So again, in all bunch of information that we have here, this protein receptor, or whatever you want to call, SEC-JAG or YEG, is important. This is a channel in the membrane. So that channel is waiting for your complex to come. Now, the other protein, sorry about that. So sorry about that. The other protein, FTSY, is like a docking for SEC-JAG. So think about all that structure on cell membrane, SEC-JAG and FTSY, which is waiting for your ribosome, protein, and SRP, all complex to come. Now, once when the complex of ribosome with protein, SRP, protein with N-terminus and SRP arrives to the membrane, this docking protein, FTSY, will recognize that this is actually the protein that needs to be translocated over the membrane. So this is a key point recognition. Now, the molecule GTP from cellular metabolism will hydrolyze and release energy. Again, this GTP can come from various different processes inside the cell. Energy from GTP hydrolysis will be used to push protein through SEC-JAG channel. So, and this is all happening, that's why it's called co-translational. And you will see in a few minutes what's the difference between co-translational and post-translational. But the key points that you need to pick from here are protein with N-terminal signal sequence, SRP on ribosomes that recognize signal sequence, SEC-JAG channel in membrane, and FTSY that is helping SEC-JAG to be on the membrane. Right. So, so let's go now to post-translational version. So in post-translational version, you have similar situation. Protein is again synthesized on ribosomes. Again, you have the key player N-terminal signal sequence that is attached to protein. So that's a part of protein here. Now, again, N-terminal is a label for specific translocation. Now, this is the difference. When protein is fully translated, post-translational translocation begins. So protein needs to be fully translated, which means ribosome doesn't migrate to the membrane. Now, there is another molecule, SEC-A, that contains a pocket that will recognize this N-terminal signal sequence on now fully translated protein. And this is still happening in cytoplasm. So you won't have ribosome that goes with the protein to the membrane. Now, SEC-A is literally carrying this protein with N-terminal signal sequence to the membrane. And in the membrane, there is again SEC-JAG channel. Slightly different situation from co-translational version. Now you have ATP binding, so not GTP. Next step is basically ATP binding to all complex SEC-JAG, SEC-A, and protein with N-terminal signal sequence. So this SEC-A, I realized as a like analog to the ribosome. So in co-translational version, you have ribosome that is traveling with the protein. Here you have SEC-A that grabs protein 1 when it's fully translated. Now, SEC-A is ATPase and cleaves ATP to ADP and PI, aka releases energy. In addition, there is a proton motive force. So in previous one, you had only GTP hydrolysis. Here you have ATP plus PMF, and those two, PMF plus ATP, are helping to push protein through the channel. Now, the first one that goes through channel is this N-terminal signal sequence. Once when you have protein in periplasm, that signal sequence needs to be removed, and that peptidase in periplasm will cleave N-terminal signal sequence, because N-terminal signal sequence is not a part of your protein per se. So you shouldn't have it in order to have properly functioned protein. So basically, N-terminal signal sequence is there just to help translocation, and then it's cleaved. And protein is released, it's a final step. Okay. Next, so this was SEC, co-translational, post-translational. Next step, next system for protein translocation is TAT, T-A-T. And here is the principle. Again, protein is fully translated on ribosome. Again, during translation, protein gets N-terminus, and that sequence is called twin arginine. That's why you have it TAT, twin arginine. After translation, protein is folded to its tertiary structure. So here you have fully folded protein. Now, player here are chaperones. So chaperones are actually molecules that are recognizing this twin arginine N-terminus and bring it to the TAT translocase in membrane. So these chaperones are doing pretty much the same thing that SEC-A is doing in previous one. So now all that protein is brought up to the translocase in the membrane, and TAT channel is a large pore. So SEC-YEG is a channel, and this one is larger pore. Now protein is very big because it's fully folded, so that's why actually channel has to be big. Once when protein is on that channel, bacteria uses proton motive force to push it to the pore outside the membrane. So this is only PMF, no ATPs, no GTPs. General structure of SEC-JAG, which is important channel for co-translational protein translocation and post-translational. So SEC-Y, which is a pore, like a channel. So SEC-G, which is surrounding a pore, like a little dots around it. and also actually sec E is surrounding a protein, and sec G is like a lid, seals the pore. And now harder part of this, secretion systems. So those tools, sec and tet, were for translocation. Secretion is a different, and in my opinion, much harder. So this part contains a lot of details. You guys should be focused mostly on lectures. I gave many different examples just for the purpose of understanding, but I will tell you what's important to memorize, to understand what's the point of each of these secretion systems. But again, for the exam, your focus are examples from Lisa's lectures, which are also given here. So for the comparison, to begin, protein secretion system can be SAC and TET independent or dependent. So if secretion system is SAC or TET dependent, that means that protein first has to be delivered to periplasm using one of those SAC or TET systems for translocation. Then, using another secretion system, protein is exported. So you need to have two components. If it's independent, then protein is just secreted through the channel that spans both inner and outer membrane, and you don't have that periplasmic step. So type 1 protein secretion system. So this one is actually really cool, and we were discussing about it recently in the lab, since one of our colleagues is discovering something very interesting regarding this system. Not my favorite, definitely, but probably one of the oldest. So general structure of T1 secretion system, or T1SS, is that it is ABC transporter in inner membrane. So if you recall what are ABC transporters, these are secondary transporters that usually use ATP to move protein through the membrane. So ABC transporter, typical way how bacteria are excreting drugs. Next component is membrane fusion protein, or MFP, which is like a connective loop between that ABC in inner membrane and structure in outer membrane. And the structure in outer membrane is outer membrane factor. This is important. So this outer membrane structure is pretty unique. It's called TOL-C. So if you think about those two membranes, outer, inner, you have TOL-C on outer membrane, you have ABC transporter to the inner membrane, and you have connective loop or connective structure in between them. Again, outer membrane factor does not have to be TOL-C, but most likely it is. It's very common among bacteria. And TOL-C is important part, so it's important to understand the meaning of TOL-C. What's the mechanism of transport? So again, when there is a substrate that needs to be exported through T1 machinery, that substrate is recognized by N-terminus of this MFP component. So that MFP is connected thing. Now, once recognized, substrate for export is binding to the ABC transporter component. So you have a binding. And again, as we discussed yesterday, binding of substrate is leading to the conformational changes in MFP. So we talked about that yesterday, binding a substrate, conformational changes, moving a substance, excretion. So that conformational change is important. So now, recognition of substrate, binding to the ABC transporter, conformational changes in MFP, and now MFP interacts with outer membrane component. So that conformational change, so binding of substrate to ABC is literally like it's transferring that conformational change to the connective MFP, and then that's transferring to the outer membrane part. That's why it's all connected in the membrane. Now, Tall C is actually the one that is the most important. Well, they're all important, but tall C is a pore where substrate goes through. How? So to move substrate through the pore, you need again energy. That's where ABC transporter comes into the game, because it is cleaving ATP. So ABC transporter usually has ATP hydrolytic activity. So it's releasing energy and pushing that component substrate through the pore. Now, type 2 protein secretion system. This is the general structure. So again, outer membrane platform or outer membrane factor, ATPase, inner membrane platform, and pseudopeels. So this is slightly different than type 1. What is the mechanism of transport? So protein for export is again synthesized with N-terminal signal sequence. And you will notice that this N-terminal signal sequence is everywhere. So this is what you want to memorize, definitely. But again, N-terminal signal sequence is not a part of general protein structure. N-terminal signal sequence is there only for the purpose of recognition and translocation. So again, so once when protein with N-terminal is synthesized, based on that sequence, proteins are targeted to the inner membrane. In inner membranes, these proteins will be exported through SAC or TAT system. So what does this tell you is that this type 2 secretion system is SAC or TAT dependent. Now, protein is in periplasm with N-terminal signal sequence. What's happening in periplasm is that peptidase now cleaves off that N-terminus, and protein remains in periplasmic space, hangs out there. Now, in periplasmic space, there is another player that comes called GSPD. When GSPD binds, this binding stimulates the activity of another protein, GSPE. GSPE is coming. And why is GSP important? Because it will bring its own ATPase activity. So GSPE will hydrolyze ATP. So when you have protein that needs to be moved, you have GSPD coming, binding. That's a signal for GSPE to come to hydrolyze ATP, and then energy is released. What's happening additionally? All these events are followed. So that's happening simultaneously with forming a pseudopilus. Like proteins are simultaneously adding because they are forming that like a pseudopore where this substrate will just move. So you think about that like two simultaneous events in periplasmic space, and the pore is formed on outer membrane space. Type 3, hard one, must be honest. So, general structure of type 3 secretion system. That's a needle. That's how it looks, like a needle complex. It has translocation apparatus, so it's complex. And there are multiple regulatory proteins, effector proteins, and chaperones. How mechanism goes. So this is mostly for interaction with immune cells. I believe that pathogens are those who evolved this type of secretion system. So bacterium is making a close contact with some of immune cells, macrophages, T cells, any of them. I'm not an immunologist, correct me if I'm wrong. Now, when bacteria makes a contact with immune cells, this contact is enhanced by adhesins located on bacterial membrane. So initial contact is happening between bacteria and immune cell, and then there are some adhesins that are, make this contact stronger. Once when that contact is established, bacteria starts to create effector proteins that are part of injectosomes. So injectosome is a complex of proteins that bacteria wants to deliver to our immune cells. It's for the purpose of attacking our immune cells. Now, finally, proteins are injected into the host cell. And here is on the example how this looks. Pseudomonas, famous bacterium. here is how it goes. So when there is no initiated contact between Pseudomonas and host, Pseudomonas keeps its T3 secretion system inactive. No need to invest energy. There are some, so in that condition, Pseudomonas is doing some weird things, and here is what's happening. So there is a protein, XE, which is accumulating in bacterial cell, and XE is binding XC protein. Now XC is trapped. And why is that important? Well, because when XC is trapped, XD is free to bind XA. When XA and D are bound, that cannot activate transcription of target injectosome genes. So basically, you have one pair, A and D, that is together, and now no transcription. Injectosome is not formed. And that is possible only if you have XE bound XC. So pair EC is preventing activation of pair AD. So this is all once when there is no contact with immune cell. What's happening when bacterium comes into the contact with immune cell? So when they come into the close contact, adhesins are expressed, and now you have like a tight connection. Bacteria attaches very tightly. All secretion is activated, and here is what's going on. Removing of pairs, breaking up. So this is a signal for E protein to stop accumulating. What does that mean? No XE, it's excreted from the cell. Now XC doesn't have its own partner, so it migrates to bind XD. That means that now XD is trapped and XA doesn't have a partner, and XA binds and initiates transcription of genes for injectosome proteins. So XA is kind of transcription factor. So this is co-regulation. Again, it's a good example just to imagine how is that regulation happening through T3 signal secretion system, because it's really tightly regulated, and I found it interesting. Even though there is no this example in lectures, I really liked it. It helped me understand better. Type 4 secretion system. Again, general structure with little variations. It's usually similar between all systems. Inner membrane, flexible stalk, outer membrane complex, and extracellular pili. And this is a figure from paper. Please don't memorize this. I mean, if you can, great. I can't. My brain doesn't function so well. And I like this figure because it shows all complexity of T4 signal system and all those Vir proteins. So Vir proteins would be the one that you might want to memorize. There are multiple. And that's inner membrane complex. Vir proteins, D, B, and different numbers. Again, flexible stalk, Vir proteins. So everything is about Vir proteins. And flexible stalk is here. Outer membrane complex, again, Vir proteins. So general structure matters here mostly. Inner membrane, flexible stalk, outer membrane complex, and extracellular pili. And that's all. Like I believe that I didn't put in details anything else. It would be too complicated. Type 5. For me, this one was complicated. General structure. Potradomain, linker, passenger. This one we never heard. So this one is special, and it's special for many different reasons. So general structure of molecules that are secreted through this system is specific. It has an N-terminal signal sequence. What does that tell you? It's Sec or Tec dependent. Passenger domain and translocational unit. What's the mechanism? So protein moves through Sec-YEG channel. Again, that tells you Sec-Tec dependent, the same like type 2. Once when protein is translocated in Sec-Jeg, the same thing, protein now hangs out inside in periplasm. So protein is fully in periplasm with N-terminal signal sequence, and protein also has this passenger domain and translocation domain. Now, translocator domain of protein interacts with passenger domain of the receptor in outer membrane. This is unique, so you have it in all systems for export is in outer membrane. Now, when this contact is initiated, like translocator domain with the passenger domain in outer membrane, protein is actually inserted. So protein inserts itself in that outer membrane. And this is possible thanks to the translocator domain, because this domain energizes itself. And I'm not sure that I understand exactly, or I don't know, is it clear how this energy is occurring, but translocator domain is providing energy for itself, and so protein could be pushed through the channel. What is important to know here is that it's a unique system, because pore or channel for translocation of proteins is in outer membrane, and there are all sorts of different variants depending on molecules translocated. And famous bacteria, which I don't know how to pronounce in English, I know how to say in Latin, so Neisseria meningitidis would be in Latin, I think, if I didn't forget. I don't know how to say it in English. But here is how this thing operates. So IgA1 is the protein that needs to be transported. So this is the protein, IgA1, that bacteria wants to secrete through type 5 secretion system. So it goes through sac-jag, and now it's in periplasm. Now in periplasm, this, so you know that there is an N-terminal signal sequence and passenger domain. So there are like N-terminal signal sequence, passenger domain, and C-terminal helper domain of proteins. That's all now in periplasm. Now, this C-terminus makes first contact with outer membrane. This is so it interacts with outer membrane, and again, insertion of IgA1 is happening. Once when it's inserted, C helper translocation or beta domain forms a channel. So it's like a self-forming. Through the channel, protein is moved out. And again, it energizes itself through C helper domain. Once when it's out, extracellular proteases complete activities in cell. So full activation is happening outside of the cell. And again, this is just example, not in your lecture, simple for general knowledge. I think it's cool to know. Type 6. This was a question on midterm. It might happen in final. I don't know yet. General structure of T6 signal secretion system is phage-like tail structure, very unique, base plate and membrane complex. So this one is fairly unique among bacteria. And here is the mechanism. So this phage-like structure initiates contact with the target cell. So it's kind of like what phage does when it wants to infect bacteria. Upon detection of target, hydrolysis of ATP by machinery component completes rapid contraction. So this ATP hydrolysis is doing contraction. That contraction is actually expelling, like ejecting cocktails of enzyme from the spike. So bacteria inside has that spike full of toxins, and when contact is made, ATP hydrolysis occurs and pushes it from the membrane. And enzymes are different depending on what's the target cell of bacterium. Like it's usually pathogenic, so it can be protease, nuclease, lipase, depending on what bacterium is doing. And the end is, of course, lysis of target cell, lysis of host cell. Type 7 protein secretion system. There is no general structure because there is nothing special there. But here are some examples. So again, this substrate with N-terminal signal sequence is folded in cytosol, and there is a protein chaperone, ECCA, which assists in leading substrate through the inner membrane. So this chaperone is grabbing your protein based on N-terminal signal sequence and leads it to the inner membrane. And this 7 machinery is in inner membrane. Now, when protein is about to be transported, the component of inner membrane, another one, ECC-C, is completing ATP hydrolysis. So think about that in this way. One protein C, ECC, is hanging on in inner membrane. You have protein in cytosol, and you have ACCA that is grabbing that protein and leads it to the C in inner membrane. Now, when protein is there, hydrolysis of ATP is occurring, and energy is released, so protein is pushed through the channel. Type 9. This one, very few examples, like I really didn't find too many. This is a general structure. It has cytoplasmic and inner membrane components, periplasmic components, and outer membrane and surface components. General structure. So pretty much similar, like almost all. The mechanism is next. So again, proteins are guided by classic signal peptides to Sec machinery in inner membrane. So this one is also apparently Sec dependent. So during translocation, the signal peptide is cleaved off by peptidase. So as protein is moving through Sec channel, that N-terminal goes off and protein is released in periplasm. Now in periplasm, transported proteins are folding. I never heard about folding in, well, it's weird that they're folding in periplasm, but yeah, okay, so they're folding in periplasm, so that's where they get stable conformation. And C-terminal domain on proteins is directing proteins to the outer membrane. So this CTD or C-terminal domain is actually sorting peptidase. Now, this sorting peptidase is cleaving CTD during translocation to the outer membrane and protein is released. So CTD is the final player that is actually determining where your protein will go. And we are done with unit 4. Let's move to the unit 5. Stringent response, stress. So what is stringent response? Let's first start with starvation when there is a nutrient limitation. First thing that you need to know is RLA, very important protein. Here is how situation goes. So when bacteria is starving, protein synthesis needs to stop. The reason for that is because cell doesn't want to spend energy. It's starving, so protein synthesis stopped. What does that mean? At a site of a ribosome, so now in like again general structure of ribosomes is something that you should know, but there is in my notes. So there is an A site at 30S subunits of ribosomes. So in that A site, tRNA that is not with amino acid, so deacetylated tRNA arrives. This is a signal, so protein synthesis stops. In that moment, when deacetylated tRNA arrives, RLA wraps around A site. So what you need to, what I think it's important to know is that deacetylated tRNA, so tRNA that does not have any amino acid, is a signal for stringent response to start, for RLA to bind. Now, RLA wraps around 30S site, and this is called stalled ribosome. So on this spot, RLA creates PPGPP from GTP. Now, transcription factor DKSA interacts with PPGPP, and now that complex DKSA PPGPP interacts with specific sigma factor. Sigma factor is part of RNA polymerase. So with that specific sigma factor, that interaction will turn on specific RNA polymerases that will activate specific stress response genes. What's happening further? Also this complex, so it's important to know this complex DKSA PPGPP, because this one, not just it starts transcription of stress-related genes or controls transcription of stress-related genes. It's also interacting with RNAP to stop transcription. It also interacts with elongation factors to prevent translation. You don't want cell to spend energy on protein synthesis. Also, you are inhibiting chromosome replication with the same complex. So this complex is the one that controls everything. This complex also interacts with promoters that will initiate amino acid biosynthesis, internal biosynthesis, because bacteria is starving, so no additional amino acids coming from the outside. Another stress response protein is called spot T. So after relay, we have spot T. They're homologs. So just the first one was nutrient limitation. I mean, this one is also nutrient limitation, but it's more like amino acids. And this is starvation for carbon, phosphorus, iron, and fatty acids. So when there is starvation for carbon, phosphor, iron, and fatty acids, spot T is activated. Usually fatty acids lacking is activating spot A. Now, spot T interacts with acyl carrier protein from fatty acid biosynthesis. So no fatty acids, ACP, the one that should carry fatty acids, now doesn't have fatty acids. And spot T binds to ACP. The result is creation of PPGPP from GTP that will regulate transcription, translation, and replication of stress-related genes. Sporulation, oh God, this one is hard. It was hard for me. So some bacteria are undergoing sporulation, and signal for sporulation is nutrient deprivation. So no nutrient, bacteria will go to sporulate. One important thing is bacteria will not sporulate if DNA is damaged. So bacteria doesn't want to save genes that are bad. Now, when there is a good cell density and no DNA damage, sporulation may begin. The first player is kinase, enzyme that phosphorylates SPOF. SPOF phosphorylated is a sign for sporulation event to continue. And you have a series of phosphor transfers. So SPOF transfers phosphate to SPOB, and SPOB transfers phosphate to SPOA. Now, when SPOA is phosphorylated, this is the main sporulation regulator. But they are all important. And all these phosphotransfers are occurring in between these proteins. FBA. So this is from what's happening. So what's happening if DNA is damaged and cell density is low? So the previous example was when sporulation will happen, where cell is healthy, DNA is good, everything is good, and sporulation can begin. So what's happening when cell density is low or DNA damage? So that means no sporulation. Here is what's happening. There is a factor, SDA, that blocks first kinase. What does that tell you? There won't be more phosphor transfers. So SPOF and SPOA are not phosphorylated. So you remember that in previous one you had kin A phosphorylating FBA. They're all kinases, they're all transferring phosphates. So SPOA phosphorylated is a sign that everything is cool. If first phosphorylation player is not active, nothing below is happening. So there are additional regulations. COD-Y GTP complex can block this PHR-ACE and ABE proteins. What does that mean? That's COD-Y GTP mediated control that participates in dephosphorylation of SPOF. So what I want you to get from this is that phosphorylated proteins in sporulation, SPO, FBA, are signal that sporulation happens. If there is no phosphorylation, sporulation won't happen. COD-Y GTP is additional upstream regulator of SPO, FBA phosphorylation. Yeah, this is the order if you went, I would pay attention on order of events in sporulation. This is important to know. The same how I would understand basic principle that sporulated phosphorylated SPO-FA is the main regulator. Sporulated happens, that means DNA is not damaged, cell density is okay. SPO-FA dephosphorylated, like no phosphate, sporulation doesn't occur, DNA damage maybe happened, or there is no enough cells, low cell density. Now, the order of events, what's happening when spore is forming in bacterial cell? Well, actually, when bacterial cell is forming spore. So first you have vegetative cell growth. So DNA is increasing in density, and septation, like division between mother cell and pre-spore, is occurring. Now, what's happening is forming the protoplasm, like enclosing the spore protoplasm. So septation happened, now protoplasm inside is forming, like parts of cell membrane are forming something inside the cell. Next one is exosporium formation, dehydration, so all water is removed from that protoplasm. Then calcium ions are incorporating. Also, simultaneously, there are dipicolinic acid that is also incorporating, pretty much same time like calcium. Next one is maturation, lysis, and release of the pre-spore. So spore first has to go through maturation, then mother cell has to lyse to release spore, and then spore is released. Adaptations. Again, this was a question on midterm. I would memorize specific adaptations. It might be a question. So pay attention on what are specific adaptations, like try to understand the nature of environment where cell is thriving, and then you will understand those adaptations. For example, when bacteria is growing in high temperature conditions, there will be specific lipids and there will be saturated fatty acids. In cold environments, again, specific lipids, different than one from hot environments, and there will be unsaturated fatty acids. Cold stress adaptation, how this goes. What is changing are fatty acid composition. So fatty acids are changing composition in membrane when there is a cold environment. Again, fatty acids are synthesized with Fab pathway, Fab AG, A, and I. And if you remember, we talked about that yesterday: carboxylation, condensation, reduction, dehydration, and again reduction. Now, even if fatty acid is saturated, enzyme desaturase introduces unsaturated bond. So even if that fatty acid is meant to be saturated because it's a cold environment, there is an enzyme desaturase, which is a key that introduces unsaturated bond. Membrane becomes rigid, and that's a signal for protein DASK, membrane protein DASK. DASK is doing autophosphorylation and activates itself. Once when activated, DASK phosphorylates another DAS protein R. And DAS-R is transcription factor that activates transcription of genes for desaturase. And that desaturase introduces that unsaturated bond. So basically, imagine this: cell is in cold environment. That's a signal. Aha, I have to convert my membrane into the rigid membrane. So to do that, I need my protein DASK. DASK is in response to the cold environment, autophosphorylates itself, and now it's active. When active, DASK phosphorylates DAS-R, and DAS-R is going to activate transcription of genes for desaturase. Now there is a desaturase that is introducing unsaturated bond in fatty acid. Acidic stress, low pH. So proton motive force generation relies on delta pH here. And what needs to be done is that cell membrane has to be highly impermeable to H+. Because if you think about this, cell is living in environment where there are a lot of protons. If everything is normal, like cell membrane is permeable for protons, protons will migrate inside and that will acidify cytoplasm and cell won't survive because there is optimal pH for each cell. So membrane impermeable to protons. Positively charged molecules like K+ stay inside the cell. That's called non-potential. Also, it has to reduce pore size in membrane channels, again, to prevent entrance of unwanted protons. There are transporters that pump out the excessive protons, because think about it, even though cell is impermeable, if something accidentally migrates in, it has to be some mechanism to remove it. So all purpose is to prevent extra protons to enter the cell. These organisms have reversed the membrane potential, so it's more positive inside than outside. Generally, it's always more positive out than in. Here is reverse. And H+ consuming respiration happens inside the cell. Again, organic acid degradation is happening to stop proton accumulation in the cell. So all what you're trying to do is to prevent too much acidification of internal environment, and also you don't want to export more protons outside. Actually, you can export more protons outside, but all point is to keep cellular environment with a normal proton level, normal acidity, normal pH level. Alkaline stress, so opposite than acidic stress. Now PMS, sorry, huh?
Can I ask a question? Sorry.
Yeah, sure.
In your previous one, talking about acid stress, with the reverse in the charge, the delta psi, is that for acid challenge or acid adaptation?
I think it's for acid challenge.
Okay, so like E. coli's in acid, it will reverse its delta psi, or is it?
So here's the thing. I believe that this reverse potential, and this is what I think. So I'm not sure. I think reverse potential is something that is happening as a response to the environment stress, like adaptation. I don't think it's genetically controlled, but I'm not sure. So don't trust me completely, because again, I don't have much knowledge in this area. All what I know is from literature that I read for this class. What do you think?
I was under the impression that was for only acidophiles, not for an acid challenge. So that's why I was like, oh no, did I get this wrong? I don't know.
I'm really not sure. Any other thoughts, guys? What do you think?
I think that it's for acidophiles as well. I think that there's a different process that happens when something's being challenged.
So I think I will agree. I think well, Jen is right. I believe that reversing of potential can happen as adaptation, like as a response to the acid challenge. So I think that E. coli, if you are constantly exposing it to the acidic stress, I think it will start reversing potential, which probably can be considered as an adaptation. Like,
okay.
That's what I would say. I know that in our lab, there was a person who was adapting methanotroph to low pH. And I think that what happened is that it eventually starts growing on low pH, even if it's not meant to grow on low pH. So I believe that in order that to happen, it had to reverse its potential. But again, this is just my opinion. I'm not convinced that that's correct. But if you can remind me, I can definitely check or consult with someone who is more from the area of extremophilic microbiology, because that's a very good question to discuss.
Thank you.
Again, alkaline stress. So you had here PMF generation relies on delta pH in acidic. Here it relies on delta psi. You have antiporter, which is going to remove sodium, and H plus has to come in to acidify cytoplasm. So all point is to keep homeostasis in cytosol. Now, so in previous one, in acidic, you wanted to get rid of protons. Here, you want to import them to maintain homeostasis. Because if you don't, and cell is in alkaline environment, eventually cell will become alkaline and won't survive. And it's sodium-based ATPase, so not proton. In high salt stress, you have a few adaptations: lower glutamate catabolism, accumulation of intracellular trehalose, and accumulation of organic osmolytes, so all inside the cell. When there is low salt stress, there are transporters that participate in that salt efflux. So when there is a high salt, bacterium is accumulating trehalose and other osmolytes to keep that osmolarity. When there is low salt, bacteria wants to get rid of those solids and exports them out. And that would be all for units 4 and 5. Any more, let's see, are there any questions?
I have another general question.
Yeah, please ask. Like, you always have great questions, and I like to hear your question because that helps me also understand and solve my problems. Say it, please.
Just for the exam, if I see N-terminal signal sequence, that's like secretat, right? Like, we know that's secretat dependent, and then I'll have to memorize all the different secretion systems, but that would then target one of the secretat dependent secretion systems, right?
If it is secretat dependent.
Right, yeah, but if it's some of the clues of the question are, oh, there's an N-terminal signal sequence, then I know I have to, if it's kind of something around like, oh yeah, how does the protein get out? Then I know it has to be one of those secretat. Like, none of the ones without secretat have an N-terminal signal sequence, right?
Yes.
Okay, just wanted to abundantly clear.
Yep. That's why I'm like pointing out N-terminal signal sequence, because that one is a marker for secretat. Yeah. So
okay, I just wanted to, I just wanted to double check because I was like looking through the other ones that aren't, and I'm like, is there anything that N-terminal? No,
you're completely right. That's how, and guys, memorize what Carmen said. So N-terminal signal sequence is sign that it's exported through secretat.
Because I know there's going to be these little clues, then we have to like figure it out with the clues and tell them what they want to hear.
And if you have that kind of clues, you will be good. There won't be a problem. And again, we will go through these all lectures by Thursday, and then on Friday and Monday we will go through the exam. God help me, I think it will be fine. I'm doing my best to help as much as I can. So you should be good. I want you to be good. Although I'm really not worried for people. Most of people here are always, not always, but most of you attended. So I'm worried for some people who never showed up. I don't know how that will go, but
well, hopefully they're listening to the recordings.
That's what I'm hoping, to be honest. Yeah, that's why I'm constantly thinking about how to upload all these videos, because maybe they will listen my voice. I mean, if they don't understand, they can always text me, email me. I can.
You can check how many views the videos get just to get an idea.
I mean, I could do that if I am model star, as I used to want to be, but because I'm not, I mean, I never even thought about having my YouTube channel. Yesterday, I realized that I actually have. I can be YouTube star. So yeah, people will laugh, definitely. So that's it, guys. We're done with units 5. Tomorrow we are going 6 and 7, right? We said 6 and 7. Yes, we will go tomorrow with 6 and 7. And I was thinking to go on Thursday, 8, 9, 10, just because unit 9 is really short and cool. And there are no many things to memorize. You can just, briefly go through it. And I believe that for you, it's easy to memorize everything from unit 2, because Lisa described that like with examples, like how microbiome is used, and it's really easy to memorize. It's like a story, like you are reading, or like when you're reading newspaper article.
Can I ask a quick question?
No, you can't. Okay.
When you talk about the toll-C in the type 1 protein secretion system, that's the same toll-C that we see for the multi-drug efflux pumps, right? So they like are part of the ABC transporters and all that. Okay.
They can be. Yeah.
So they can do both protein secretion as well as like drugs. So not necessarily just okay, cool.
Yeah. Sometimes some drug can have protein structure, like amino acid structure. So yeah.
Okay, cool.
You got it.
Great.
Are we good? Excellent. See you then tomorrow. I will check out. I'm going to eat something. So, and to convert this to YouTube, so you can all see it later. And you have all lectures posted. I mean, Word document, everything is posted. PowerPoint, I didn't finish everything. Hopefully, I will finish it by tonight, if not by tomorrow morning for sure. And see you tomorrow, same time.
Great.
Thank you. And thank you for coming, guys. See you. Bye.
Thank you.